The epidemiology of dilated cardiomyopathy in adults and children is somewhat different. So the incidence of heart failure is much more common in adults than in kids. But the consequences are as bad in kids. So in 5 years, there's 50% death or transplant in the pediatric population. In adults, the primary etiology is ischemic heart disease, whereas in kids it's very heterogeneous. It's congenital heart disease, dilated cardiomyopathy. But the difference is that kids don't have a lot of comorbidities like adults do, like smoking or alcohol. It doesn't play a role in pediatric heart failure. They are considered a vulnerable population, so it's harder to do those studies. There are not so many people working on this field. But like I said in the beginning, one of the problems is this assumption that children should respond to the same therapies as adults. So there are no strong evidence of any therapies for pediatric heart failure, so 0 level A recommendations for pediatric heart failure. And unfortunately, because of this assumption that kids and adults were similar, you can see here in these graphs that the outcomes in kids hasn't really changed. So this is adults here on the left-hand side. This here is a historical cohort. So before the more common use of therapies such as beta blockers and ACE inhibitors, this is the more contemporary cohort, and in adults, you can see that all-cause mortality has decreased dramatically between these two cohorts. Kids are here. This is 2 years in adults, this is 2 years in kids, and even if you go longer, it doesn't really make a difference. The point here is that if you look at the perspective, which would be the same therapies that are used in adults, compared to the retrospective, which is compared to this graph, this line here in the adults, there really hasn't been an improvement in outcome in the pediatric population. So this improvement has been minimal, even though the same therapies are used. Like I said, in 2007, this study was published with carvedilol for children with heart failure, and they did not see an improvement. And this included congenital heart disease, dilated cardiomyopathy. It was all comers of heart failure. Interestingly, though, PD3 inhibitors, which are known to cause increased chances of sudden death and arrhythmias when it's chronically used in the adult heart failure population, in kids, it's very well tolerated. Kids can go home on an inotrope. They can have a line put in, they're fine. And there have been these studies published that this is probably a good alternative to treat kids with heart failure. I'm not going to show the data that we've published showing that the molecular mechanisms of why carvedilol probably didn't work in the pediatric population and why a phosphodiesterase 3 inhibitor works in kids and the mechanisms of tolerance in adults. So that's published data, and I want to focus more on newer work in the lab. So what we try to do in the lab is go from the patient to understand the mechanism. So like I said, we have the advantage of having this tissue bank, and we also collect hearts as they are explanted. We study mitochondrial function. We've done a lot of work with microRNAs. Like I said, the phosphodiesterase system. We've now looking at metabolomics and lipidomics. We've done transcriptomics of those hearts. And then myofibro and myocyte mechanics. There are 2 junior faculty in our group that are focused on those studies. We've also, because there are no good animal models of pediatric heart failure, we developed a model that uses paracrine factors. So we use serum from these pediatric patients to treat cells to try to understand the disease process. So just for background, we show, this is also published work, but we show that fibrosis, like you see here in adult heart fibrosis is very common in adults. In kids, fibrosis is rare, especially interstitial fibrosis, which is more common in adults. We really don't see much of that in kids. We also did the transcriptome profile of pediatric patients. And what we saw, and again, this was a little surprising, was that there are fewer genes that change in the pediatric dilated cardiomyopathy heart. So this here is kids, and this is upregulated genes. These are the downregulated genes, and this is kids. This is adults. But importantly, of the genes that change in the pediatric heart, most of them change. The changes are unique to the pediatric heart. Again, pointing out to age-specific differences in pediatric heart failure. And then we looked at cardiomyocyte hypertrophy, and this work was done by Caitlin Wolf. And what we see here is that adults, as known, there is an increase in cell size. Hypertrophy of the myocyte is common, and you can see here and here, it's measured here. And then in the pediatric heart, we actually did not see an increase in cell size. So hypertrophy is not a characteristic of the pediatric heart. So again, we wanted to understand the heart. We don't want to use models of heart failure where the animal is sick when they're young. We actually want to understand what's happening in the human to then develop these models. I'm not going to talk about our in vivo work right now, but I'm going to focus on the in vitro work. So what we have been doing is using the serum of kids that have heart failure, dilated cardiomyopathy. That's the only, we also work with single ventricle heart disease, but I'm not going to talk about that work. I'm only going to talk about dilated cardiomyopathy. And we treat NRFEMS because we've seen that NRFEMs reproduce well the characteristics of the pediatric heart. And we look at pathologic remodeling. We've looked at circulating microRNAs as causative factors in that pathologic response, circulating proteins, and I'm going to focus on SFRP1. We've measured cellular stiffness, mitochondrial function, lipid remodeling, and metabolomics. So what we do is that we use the serum from these pediatric patients, and all of our comparisons are to cells treated with serum from kids that don't have any disease, so non-failing controls. We have published with NRFEMs and iPS cardiomyocytes, and we've shown that for those measurements and that work was focused on gene expression changes, that the response in NRFEMs and iPS cardiomyocytes was similar. This work was done by Sean Jiang when she was a postdoc in the lab. She's now at UT Southwestern. And what she showed here, this is in NRFEMs. So again, treating the cells with serum from non-failing or dilated cardiomyopathy patients, we see an increase in the gene expression program that is associated with pathological remodeling. So the next question was, what is causing this? What factors in the serum? And what Sean looked at the time was if exosomes were involved. So exosomes can carry a lot of nucleic acids and proteins, and they are known to involve this, produce this paracrine response. So they're involved in cellular communication. So when she isolated exosomes from serum and treated the cells, the exosomes, which are these, the left hand side of the graph, did induce that response, so increasing in ANF, BNP, and beta-myosin heavy chain, but the supernatant from those exosomes also had a response. So it looked like there were more than one factor that was causing that response, some present in the exosomes and some present in just the serum. Surprising, though, to us was that when she looked at the amount of exosomes in the pediatric serum, different than other diseases, especially cancer, where you see a much increased number of exosomes. In kids with heart failure, we actually saw fewer exosomes. So this is non-failing, and this is the CM. This is a pictures of the nanocyte, and we see fewer exosomes from the serum of these pediatric hearts. In association with that decreased number of exosomes, we also see fewer circulating microRNAs. So this is the volcano plot. So everything that's green and above the blue line is downregulated, and each one of these dots is a microRNA. So all of these microRNAs are downregulated in the serum of the dilated cardiomyopathy patients, and only one of them was upregulated in dilated cardiomyopathy patients. So we wanted to know because our hypothesis in the beginning was that microRNAs were causing this, so that there would be more microRNAs and they would be causative. So that's because they're generally increased and they can act as paracrine factors. So to understand this, what we've done is that we've developed a methodology to kind of release these microRNAs in the serum so that we can understand their function. So, and this has been published. So we release these microRNAs and then we treat them with either RNAs or DNAs, or we treat the serum, not the exosome content, with proteinase K also. So, but the problem is that if you don't have the exosomes anymore, there's no guarantee that what was present in the exosomes is actually going to affect the cells. So we transfect that resulting serum so that we can see the response. So because there were fewer exosomes than at that, and fewer microRNAs, we thought that maybe the microRNAs would be protective against pathologic remodeling. And that's what we saw. This work has been mostly done by Danielle Jeffrey, who is starting her PhD this fall. So again, what I'm showing here is BNP, ANF, and then the ratio of alpha-myosin heavy chain to beta. And if you look at just a non-failing serum treatment, and none of this work has been published, if we treat the serum with DNAs or RNAs, we see with the RNAs, we see an increase in the pathologic response if the non-failing serum is treated with RNAs. So that suggests that the RNA that's present in the non-failing serum is actually is protective and preventing the disease response. So this is BNP and NF, and there's a trend in the alpha to beta myosin heavy chain ratio. When we treat the cells with serum from the DCM patients, again, we see this pathologic response, but neither treating the serum with DNAs or RNAs really changes any of that. There's a decrease here in ANF, but this suggests that the response that we are seeing is really not mediated by RNA or DNA. However, when we treat the serum with proteinase K, what we see is a complete reverse of the pathological gene program. So this increase in BNP in response to the DCM serum goes back down when we treat the serum with proteinase K. Same with ANF, and same with the alpha to beta myosin heavy chain ratio. So this strongly suggests that the causative factors here are protein mediated. So we wanted to know what was causing the disease in the cells, this pathologic remodeling. So we did a transcriptome analysis of the NRFEMS that are treated with serum from pediatric dilated cardiomyopathy patients. And this work has been a collaboration between Danielle Jeffrey, Julie Perez da Silva, who's a postdoc in the lab, and Anastasia Garcia, who is now an assistant professor in the division. And what I'm showing here is that the serum from these patients results in a very different expression of genes, as you can see here from this heat map. So it's very unique to the response to serum is very unique. And what I highlighted here, this is IPA analysis, IPA and Metascape. What I'm highlighted here is in red are what I'm going to talk about and discuss in this talk, which is mitochondria studies and cellular stiffness that came up as factors that are probably affecting this response. So to look at cellular stiffness, we did studies with atomic force microscopy. I don't have the time to go into the methodology itself, but it allows us to see how the cell responds to a pressure, and that allows us to measure stiffness. So when we treat the NRVMs with serum from these pediatric patients, we see an increase in cellular stiffness. At that point, we already knew that proteins were what would probably be causing this response. So we used the somologic SomaScan methodology to characterize the proteins that are present in the secretome of the pediatric dilated cardiomyopathy patients. And what I highlighted here is that the focal adhesion signaling and ECM remodeling is because that was what is very similar to what we saw from the transcriptome of the NRVMs. So we wanted to know what proteins that are present in the serum that could be causing this response. So one of the proteins is SFRP1. SFRP1, this is the circulating factors here. So the amount of SFRP1 that is present in the serum of the pediatric patients, and we see a dramatic increase in circulating SFRP1 in these patients. And previous studies have shown that SFRP1 can increase cell stiffness. It can also be involved in inducing apoptosis. It's also involved in senescence. And in human heart failure patients, pre and post-VAD, they saw a decrease in SFRP1 levels that were associated with recovery. So to us, that there are some other studies that have shown that SFRP1 can be protective. We do think that it may be one of those. If it's circulating, it has an effect. If it's endogenous, it has a different effect. I'm not going to go into any of that. But the interesting thing is that some of the microRNAs that we know are downregulated in the pediatric dilated cardiomyopathy heart are predicted to target SFRP1. So it's possible that the fact that these microRNAs are downregulated can be contributing to the effect of SFRP1. So when we look at the pediatric tissue, the LV tissue, we also see an increase in SFRP1 in the tissue of these patients. And again, doing AFM, and this work was done by Brisa Penny-Castellanos, who is also an assistant professor in the division. When we treat the cells with the NRVMs with SFRP1, we also see this increase in stiffness. So this suggests that at least when we talk about the ECM remodeling that we are seeing, that SFRP1 may be a factor that's causing this increase in stiffness and may be involved in ECM remodeling. So I'm going to talk now about the mitochondria studies that we've done. This is truly a group effort because it does take a lot of time to perform these studies. So here the cells, the NRVMs, were treated with, again, serum from non-failing or dilated cardiomyopathy patients. And using Seahorse, we measured mitochondrial function. We also measure production of reactive oxygen species. So what I'm showing here is that mitochondrial function is down. So here's basal respiration, ATP production, and coupling efficiency. They are all down in cells treated with serum from these dilated cardiomyopathy patients. ROS is not significantly up, but it's trending up. We haven't been able to do too many experiments since March. And this is published work. This was Katie Chatfield, who is an assistant professor in our group, was the first author in this paper, and then Brian Stauffer was the leading investigator in this paper. And this is from freshly explanted pediatric and adult tissue and non-failing controls. So we get non-failing controls when the hearts cannot be used for transplant for some reason. So what we're showing here is that in the tissue of these patients, we do also see a decrease in mitochondrial function measured by O2 flux here using a Ouroboros O2K. So we were interested in finding out what could be causing these decrease in mitochondrial function, both in the cells treated with serum and in the hearts of these patients. So one of the things that we looked at were the lipid remodeling. So Genevieve Sparagna, who's also faculty in the division, she is a lipid expert, especially cardiolipin. And so she did an evaluation of lipid remodeling in both the pediatric and adult heart and in the cells treated with serum. So as you can see here, phosphatidylglycerol is decreased in the pediatric heart, and it's also decreased in the cells treated with serum. PI we only saw in the heart, we didn't see in the cells. But cardiolipin, which is a lipid that's very, very important for mitochondrial function, especially in the heart, was decreased in pediatric tissue and also in the cells treated with serum. Cardiolipin has a lot of roles in cellular function, and so it's involved in maintaining the cristae. It has functions in apoptosis, oxidative phosphorylation, import of proteins, fission and fusion. And so one of the lipid forms of cardiolipin, which is the 1448, one of the species, is very important for, and it's the most common one in the heart, and it's very important for proper cardiolipin function in the heart. And it's decreased in the pediatric DCM heart, as you can see here, but it's also decreased in the cells treated with serum from these pediatric patients. So remodeling of cardiolipin involves several genes. And these are the ones in red here, the ones that we saw changing. And so it's through this remodeling that we get to this final species, which is the 1448, which is enriched in linoleic acid. So we then looked at this work was done by Julie Perez da Silva. We looked at the expression of these genes in response to serum from these pediatric patients. And we saw a decrease in expression of the genes that are involved in this cardiolipin remodeling. So again, there are factors in the serum that are inducing that response. So then, because this is all very related to metabolomics response, metabolism, we did metabolomics of the NRFEMS treated with serum from these pediatric patients, the serum itself to see which metabolites are changing, and of the tissue of these pediatric patients. And what I've done here, and these different color schemes highlight the differences or the commonalities between them. So everything that's in red is changing in all of them. Everything that's in green is just between serum and tissue. What is in blue is between NRVMs and serum. And then I highlighted here that in NRVMs, one of the top ones was exactly the linoleic acid metabolism, which is involved in that remodeling of cardiolipin. So I am going to focus on the fatty acids, and this is super new data, like last week data. So we haven't had a chance to show that these do have an effect, but what I did is I put together what we are seeing with the metabolomics and compared to what we are seeing in the heart. So I'm going to talk about a little bit about the fatty acid processing and ROS. So one of the metabolites that is seen increase in the serum is pantothenol, and pantothenol is known to inhibit the processing of isocarnitines, and that then switches to a glucose metabolism, which is known to happen in heart failure. So in the NRVMs, what we saw, and I think that what we are seeing in the NRFEMs is a initial response to the disease where the tissue is the consequence of the disease. So what we saw with acylcarnitines in NRFEMs is an accumulation of these acylcarnitines, most likely because the processing is not happening. In tissue, what we see is a general decrease in carnitines. And then we also looked at factors that are involved in ROS production. So this is in the serum, both of these. Glutathione cysteine is a precursor of glutathione, which is prevents this ROS, and it's important to prevent ROS. And hypoxanthine actually induces the reactive oxygen species response. So we saw that increase in the serum, and we saw an increase in the cells treated with serum. And again, we see an increase in ROS in these cells. So we think that these metabolites in the serum are playing a role in inducing this ROS response through also modifying the metabolites that are present in the cells treated with serum. And finally here, this is glutathione again, which is a ROS inhibitor. It's decreased in the tissue of the pediatric DCM hearts. And in collaboration, I didn't show this data because it's still very preliminary, but we are looking at ROS production in freshly explanted tissue from these pediatric patients. So I just have the conclusion slides. So, what I've shown is that serum circulating factors are important for this pathologic remodeling, that we actually have lower levels of circulating microRNAs, and we think that this lower level of circulating microRNAs contribute to pathologic remodeling. So it's because the circulating microRNAs are protective. That increased levels of SFRP1 in the serum promote stiffness and pathologic remodeling. And that factors that are present in the serum alter lipid remodeling and also negatively affect mitochondrial function. So a lot of what we are seeing in the pediatric heart can be reproduced by treating cells with dilated cardiomyopathy serum. So what we're trying to do is, by identifying these factors, then identify new therapies for this population. And I mentioned several people that that have contributed to the work here. Like I said in the beginning, Brian Stauffer and Shelly Miyamoto, we've been working together for the past 13 years. I couldn't have done any of the work without them. We really do everything together. These are the faculty in our group, and I mentioned the work of several of them. Stephanie Nakano was instrumental in the beginning when we did the studies with phosphodiesterase 3 inhibitors. She's now doing a lot of work with the single ventricle population and myocyte mechanics. Katie is very involved in the mitochondria studies with Jen. Casey Wolf is doing the myofibro studies, and Tasha Garcia is involved in the single ventricle population. She's very interested in the metabolism in this population. Postdocs in the lab, I showed the work of Julie Pires da Silva. Frey Hoilu has been working mostly on microRNAs, and I didn't show any of that work, and I failed. Tobat Chikato just joined the lab as a postdoc also. And these are past lab members. I showed the initial work that was Chuan Zhang was involved in. And the PRAs, like I mentioned, Danielle is starting graduate school, and then Chris and Raleigh have been instrumental in all of the mitochondria studies. These are our collaborators. Matt Taylor and Luisa Mastrani have been close collaborators. They are the ones that are studying the transcriptome work in the adult hearts. Briza and Thomas have done the AFM work. I could not do any of this work that I presented without Inez. She's our bioinformaticist collaborator, and she's amazing. She's the one that does all of those studies. So, and this is our funding, and I'll be happy to take questions.
Well, thank you, Kika. That was really, really nice talk. And I mean, you can probably stop sharing now.
Yeah.
There we go. Perfect. Really nice talk. Thank you for lots and lots of unpublished data as well. Good luck with the publication. I hear they're being written up as we speak. So there's lots of questions already. And I will start with Emma Robinson. Hi, Emma. Emma's one of our regular attendees here, probably one of the most engaged. I'm not giving any hints, but you know. Thanks, Kika. You mentioned towards the start of your talk the major transcriptomic differences between adult DCM and pediatric DCM is basically a lack of myocyte hypertrophy-related genes in pediatric DCM patients. Yet in your neonatal cells, you see ANF, BMP, and beta-myosin heavy chain activation, which is somewhat the pathological hypertrophy signature. Did you see cellular hypertrophy in your neonatal cells?
So thank you for the question. I should have said that we do see pathologic remodeling of these same genes in the adult population. This has been published for a while and didn't show any of that. So we do see an increase in ANF. We do see a decrease in the ratio of alpha to beta myosin heavy chain in the hearts of these patients, even though they don't have hypertrophy. We do see a minor hypertrophy in the cells treated with serum. My hypothesis, and I don't have any evidence for this, but what I think is happening in the pediatric heart is that they kind of bypass that compensatory phase. So, if you think about heart disease, you have that initial insult that will then produce that pathologic hypertrophy, and then that moves into heart failure. Fibrosis and all that, it's all part of that initial compensatory response. I think kids bypass that and they go straight into heart failure. So they do have the pathologic remodeling when we talk about gene expression, especially the fetal gene program, but they seem to skip this other aspect of the pathologic, the compensatory phase. The progression to heart failure is pretty fast. It's usually less than a year they need to be transplanted.
Great, thank you. So Emma's got another question. Do the pediatric dilated cardiomyopathy patients have developmental structural defects at all?
No, not that has been. It's not a congenital disease process. It is really truly idiopathic dilated cardiomyopathy. So in terms of the failing heart, it does look like an adult failing heart, but it's not a congenital heart disease. So no developmental structural defects. Not in this population. We do work, like I mentioned, with the single ventricle disease population. These are kids that are born with one ventricle only, and then they eventually develop heart failure, but that's a different population altogether. It's interesting, I'm just going to say that it's really interesting to me because actually the kids that have heart failure, because they have single ventricle heart disease, their profile in terms of changes in gene expression and all those other characteristics are more similar to adults than they are to the pediatric dilated cardiomyopathy patients. So it's not an age dependent response.
Thank you. So we've got a question from an anonymous attendee. In the pediatric cardiomyopathy hearts, have you noticed any mutations in mitochondrially derived RNAs? And if so, how frequently? And then there's a couple of questions after as well. We can go to them after you answer perhaps this one.
Yeah, so we have not looked at mutations in mito-derived RNAs. Actually, we have not looked at mutations specifically, even on nuclear DNA. So I should add that Matt Taylor, who is one of our collaborators, he was awarded a top med, which is an NIH grant that is allowing us to DNA and RNA sequencing our bank, so adults and pediatrics. So that's ongoing right now. We don't have that data yet.
Well, I actually know Nathan Tucker is online as well watching. So you never know. Maybe you could send some over to him as well. Not that I'm trying to say that. Right. And the second part of the question was in the volcano plot, pediatric DCM part, right side. Could it be the less exosomes are released, resulting in less microRNAs detected And did you control for equal RNA content?
Okay, yeah, so I think that's a very important question because our results are different than what other people have published. We purposely did not control for amount of RNA when we did these studies. So this, like I said, this is a methodology that we developed in the lab. We use only 3 microliters of serum to produce the microRNA quantification that I showed you. So we put the serum through this heat freeze process to release the microRNAs from vesicles, but we don't quantify it. What we've noticed with quantification of small RNAs, small amounts of RNA, even when we do it from extracting RNA from serum, is that the quantification is not precise at all. So we discussed this a lot with Thermo Fisher, who we used their arrays, and in the end we decided to go with volume only. So it's all by volume, it's not by amount of RNA. If we had done by amount of RNA, I don't think we would have seen this difference in this decrease in the levels of microRNAs, because we would have corrected for that. And that's the one thing we are trying to avoid, because we don't want to correct. We want to see how much is there in the serum of these patients. So we do not correct for the amount of RNA.
Great, thank you. So we have a question from Rossana Bassani in Brazil, I believe. So thank you, Kika, for this interesting talk. You have not found myocyte hypertrophy in pediatric heart failure patients, but have you seen any increase in cell density or proliferation instead?
So we are currently looking at that. I don't have an answer for you. From the preliminary studies that we have done, I would say no, there's no indication of proliferation. What seems to happen, and this is very, very, very preliminary at this point, but what seems to happen is that they have other cells that are present there, and I don't know what these cells are. They don't stain for alpha-actinin. They are not fibroblasts. So there seems to be an infiltration of some other cells or proliferation, and maybe of some other cell types that are not cardiomyocytes. But like I said, this is super, super preliminary at this point.
So why did you say they're not fibroblasts? You mean, did you stain them with sort of fibroblast markers like Vimentin, etc.?
So the interesting thing is that they are all positive for Vimentin, but they are not positive for PDGF or any of the activated fibroblast markers. So I don't know what they are. And like I said, I mean, these are difficult studies because they're very time consuming, and we do them in collaboration with the immunology core at the university, that they have a vector system that allows us to stain 6 different antibodies for each slide. And we've done some adults, we've done some kids, and now in the process of sending another 100 samples to them. So that's why I say it's preliminary at this point.
But I guess you've not seen any fibrosis anyway from your data,
no.
So it kind of makes sense, perhaps. Fine.
Correct.
Great. Okay. So next question is from Saurabh Mandal. It's got 2 questions. So do you only detect SFRP1 to be upregulated in the serum? Or do you also see changes in other family members such as SFRP2, 3, 4, and 5?
So this analysis was all done based on the SomaScan. At the time we did the SomaScan, there were a little over 1,000 proteins that were detected in the serum. I would have to look at the data, but I think that the only 2 family members that we detected were SFRP1 and SFRP5 that were detected using the SomaScan platform, and SFRP1 was the only one that was upregulated. But I don't think it detected any of the 2, 3, or 4, but I really would have to look at the data. But even if they were detected, they wouldn't have been upregulated, but I am not sure.
Right. And the second question, well, I'm not actually sure if anybody can answer this properly, but it's an interesting question. So recent studies describe the effect of various microRNAs on reprogramming of fibroblasts into functional cardiomyocytes. What is your view about such microRNA-mediated directory programming strategy for heart failure at a clinical level in order to achieve increases in cardiac functional outputs?
I mean, this is a pretty controversial area, and there are people that think that that is a way of promoting proliferation and converting fibroblasts to myocytes. This is not something that we have looked at all, and even though our circulating microRNA profile shows all these microRNAs being downregulated, when we've done RNA-seq, and that's the work that I did not describe from pediatric heart failure, and then it's a very different profile. We do see a lot of microRNAs being upregulated in the heart. So it's difficult for me to say, because, and this is not myocyte specific, that would also affect fibroblasts because it's heart tissue, it's not single cell. So I don't think I can comment to how the work that we are doing would inform the reprogramming of fibroblasts into functional cardiomyocytes.
Yeah, tough question to answer.
Yes.
Almost impossible. Right, anonymous attendee asks, could you elaborate a little? What is the current dogma about relationship between the ROS and the exosome release in the dilated cardiomyopathy tissues of serum?
So we haven't not evaluated if ROS induces the release of exosomes. What we've done is look at the serum treatment and see that increases ROS. So, and again, like I said, our preliminary studies do suggest that the ROS is increased in the tissue of these pediatric patients. If that has a role in the exosome release, I don't know, because I have to say that the ROS seems to be increased in both the peds hearts and in the adult hearts. So if there's one thing to this day that we have not seen differences between pediatric and adults is with respect to mitochondrial function and with respect to ROS generation. So those seem to be pretty similar between kids and adults.
And we have another question from Rosanna. Have you seen comparable results with the neonatal right ventricular myocytes and induced pluripotent stem cell derived cardiac myocytes? So
what we've seen with respect to changes in gene expression, I didn't show that data because it's been published, and then we focused on the NRFEMS only. The results that we have in the lab do suggest that, and this is not our result, this is actually a collaboration with Dr. Kunwa Song's lab, because they have been looking at, he's the cardiomyocyte, the IPS cardiomyocyte expert in the division. They've been looking at IPS cardiomyocyte maturation with respect to mitochondrial function. And what we've seen with cardiolipin composition and all that is that NRFEMs are more similar to a human heart than the IPS cardiomyocytes. The IPS cardiomyocytes have to go through several steps of differentiation to get to the similarities to the heart. So that's why also we stopped doing work with IPS cardiomyocyte, because the NRFEMs for what we are studying made more sense to evaluate. So it's only in the beginning that we looked at IPS cardiomyocytes.
Okay, but have you, I mean, there are some people that obviously drive IPS-derived cardiomyocytes to a more adult phenotype using, various methodologies. Thomas Eschnagen springs to mind, etc. And I just wonder, have you ever actually tried to use, the cardiac myocytes which are slightly more adult-like, IPS-derived, or were these just commercially sourced ones, which obviously are quite immature?
So when we did the work, we did in collaboration with Joe Wu at Stanford. So we got the cells from him. We did not take them to maturation at all. It was very much in the beginning. The work that Kunwa Seng's lab is doing is mature. So they are inducing this maturation profile to try to get to what the mitochondria would be more similar to in a human heart. But that requires, like I said, several manipulations, and it takes a lot of time to get the myocytes, the iPS cardiomyocytes to become mature. And, for us, the NRVMs are, it's a lot easier to get to what we need to study.
I see that Joe Wu is also watching, so so he may want to comment as well if he wishes. Right. So the next question is Chi Kung Lam. I apologize if I'm mispronouncing it. Are there any is there any mutation specificity towards the serum findings, or do the findings explain the convergence of final etiology?
So this is speculation at this point. What I think, and I mentioned that during the talk, what I think we are seeing is that what is being released in the secretome from the hearts of the patients has the pathologic factors that we can then reproduce some of the phenotypes in the NRVMs. But I think what we are seeing is the initial response to the cardiomyocytes to that insult. We have not looked at any mutations that, like I mentioned, that is being now part of the TopMed grant that is looking at the mutations, that trying to identify mutations. But no, I should also say that the patients that we have been studying are they are not familial dilated cardiomyopathy. Those are sporadic. So I am sure there are mutations. I mean, there have to be, right? There has to be mutations to be causing this, but it's not like it's a hereditary response. So I don't know the answer to your question.
Yeah. And then another question from Kumaravelu Jagavelu. Again, apologies if I mispronounce this. Great talk, Kika. Did you inject the fatty acid, I suspect this is cardiolipin, in vivo to check the reproducibility? I might be wrong though.
So yeah, I don't know exactly. No, we did not. Short answer is we did not inject fatty acid. When we look at medium chain fatty acid profile in the serum, we really didn't see much. So it doesn't seem that it's the fatty acid in the serum that is inducing those responses, but it's the remodeling of the fatty acids in the cells that is inducing that response.
Okay. And then from Alicia Mattiazzi. Hi, Alicia. Thanks for the interesting talk. Do you know whether there are differences in calcium mishandling in heart failure, pediatric myocytes versus adult?
So we published that, and I don't remember the answer to that question. Sorry, I really don't. It is published. Stephanie Nakano is the first author, but I'm really sorry. I don't want to say it because I don't remember what the...
Well, Alicia, maybe you can contact Kika and she can send you the reference as well. But actually, Kika, I mean, are there differences in beta receptor expression between the pediatric and...
So not in the non-failing. The non-failing hearts, the beta-adrenergic receptor is similar, so we see beta 1 and beta 2. The difference is in response to heart failure. So what we've seen is that in response to heart failure in adults, the beta 1 is desensitized, but the beta 2 doesn't change. What we see in the kids is that both beta 1 and beta 2 go down. So what we think, it's not that we don't think that beta blockers are not a good therapy for pediatric heart failure, but the choice of carvedilol for the clinical trial was probably what was problematic, because it inhibits both beta 1 and beta 2. And if anything, I didn't show any of these data either, but if anything, we think beta 2 is protective. So we think that if the choice had been metoprolol, which is a beta 1 specific, versus instead of carvedilol, the results of the trial would probably have been different.
Okay. Right, so we've got Charles Chung. Charles Chung is actually the last presenter in the cardiovascular webinar series. Dr. Sucharov, very nice and fascinating talk. I have two questions. Does your serum work show any clear overlap with the Python postprandial hypertrophy? which was also related to blood properties. So perhaps let's go for that one first and then the second one afterwards.
So I think you were talking about the work from Leslie Lyman's lab. So Leslie was actually my mentor when I was a postdoc. The work that they showed there was a physiologic hypertrophy in response to the Python serum. So, and that was because the animals were fed and the hearts get really big. And they saw a lipid, as far as I know from the last that I've heard, it was that it's a lipid response. We are looking at pathologic factors, so I don't know if, and they see a pretty dramatic hypertrophy. We don't see a pretty dramatic hypertrophy in response to serum.
Thank you. So, and the second question, I guess, relates to this work that you presented today. If I recall properly, you showed a change in fibrosis-related genes. Any thoughts why these are not causing interstitial fibrosis?
So, the changes in fibrosis-related genes are not the same as adults. So this is, some of this work was published and I didn't show it. What we see is when we treat the cells with the serum, we do see an increase in collagen genes and genes that are related to fibrosis. We don't have fibroblasts there. This is just cardiomyocytes. I think that this is similar to the hypertrophy question from before. I think that they just progress very fast into heart failure, and that's why we're not seeing fibrosis. Pure speculation, though.
Okay, and then the next question is from Huda Cohen. I guess it relates to one of the earlier questions, but thank you, Dr. Carmen. Do you have any idea about the etiologies of DCM the serum was taken from? Is it acquired or is it congenital?
So neither. It's actually, these kids are, it's sporadic. Like I said, it's not congenital. They're mostly idiopathic. We don't know the reason they have heart failure. They develop dilated cardiomyopathy. Kids are a little different than adults. The cardiomyopathy in kids. So kids a lot of times are, even though their ejection fraction may be pretty low, they still are running around and exercising and going to school and all that. So that response is different. Its etiology, I would say, is idiopathic. We don't know the cause. It's not familial, like I said. And as far as we know, there are no inherited mutations, at least from what we know right now. So none of these kids have mutations in the typical sarcomeric genes that have been well characterized in adults.
Great, thank you. So another question which sort of replicates one of the earlier questions, I think, is from Kaushiki Mahapatra. Any pointers of neonatal cells on iPS-derived cardiomyocytes? I suspect, are there any similarities probably in responses? That's how I interpreted. So
like I said, we really focus on NRFEMS because of these differences, especially when it comes to mitochondria composition, cardiolipin composition in the iPS cardiomyocytes. So that's but we don't work with iPS cardiomyocytes, so I'm not the best person to answer that question.
And Nathan Tucker says, I'm here for collaborations, I suspect, as well as announcing the presence. So Yuan Zhang, very interesting talk. Did you observe the changes in glucose metabolism in your Metabolomics data set, especially pediatric heart failure?
So we do see what suggests to be a switch from fatty acid to glucose metabolism. Like I said, the carnitines are almost all down in the hearts of these pediatric patients.
Great. And Nathan again, wonderful talk. And apparently I somewhat hinted at his question, but are there efforts to identify differences in cell composition versus cell state at a single cell level? He's curious what you use as a baseline to assign cell identity using a multiplex staining approach with regards to the immunology people, okay, or compared to the immunology.
So this is I don't know if this is the answer that you're looking for, but we have developed now several panels with immunology people to try to better understand what is the cellular composition of these pediatric hearts. I think we are investigating 25 different antibodies that would characterize these different cell compositions. So that would allow us to try to better understand what is happening in the heart. With respect to your question, the cell state at a single cell level, that's difficult because these are frozen tissues. So there's just so much that we can do. So I don't know if that answers your question. I'm curious what it uses as a baseline to assign cell identity. I mean, I don't know if that's the markers, but I don't remember them all off the top of my head. So alpha-actinin is there, of course, and PDGF, vimentin. There are markers of endothelial cells, of activated fibroblasts, immune cells, but I don't remember them all off the top of my head.
Well, perhaps Nathan, you can get in touch with Kika and then discuss this offline as well. So next question is from the anonymous attendee. Thank you for the nice talk. In the serum, did you measure ketone levels, or were changes in ketone metabolism shown in Metabolomics data?
Yeah, so that's interesting. We do see some metabolites that are related to the ketone metabolism, and I don't remember. I actually had written that down because we are looking at this data. I mean, like I said, we just got these data, and I can't remember, but I think there's some downregulation of at least ketone precursors in the serum metabolomics.
Thank you. And Nathan, again, we can't get rid of him. Follow up. No, Nathan, please keep asking the questions. Follow up to the inheritance question. Are there trio-style genetics analysis of these patients to identify de novo pathogenic variants? Relatively low.
So
this might be an interesting approach.
Yeah, so the short answer is no. For the majority of the patients, that no, we don't. It's complicated because this is not a familial situation, so we don't have a lot of times we don't. And these are patients that came to Children's Hospital once they already had the disease. It's not a family that we have been following or anything like that. So a lot of these, and this has not been the focus of our work also. There are other people that are doing trios. And so we are not looking at the trios. I think you skipped the question about the proliferation.
Did I?
We haven't looked. Have you checked the proliferation of NRFEMS? It's a question from Hong Chao. Whoa.
Oh, that's, you see, for me, it's the next question.
Oh, really?
For some reason. I don't know why.
Oh, interesting. Okay, sorry.
No,
we have not. We have not looked at proliferation, no.
Fine. And then we have another question from Huda Cohen in relation to a previous question. So thank you for responding. So not the secondary DCM to viral myocarditis.
Correct. We try to avoid including patients that had proven myocarditis.
And last question is from anonymous attendee. Did you by any chance check any proliferative capacities by the controlled DCM serums on neonatal right ventricular myocytes? And there's a follow-up question, plus and check for any signs of apoptosis.
Yeah, there's no evidence of apoptosis. Looking at the serum, I wouldn't say that we have not looked to see if they are proliferating or not, like I said before, but looking at the cells and following these cells over a few days, it doesn't look like we have more cells. I don't think there's any signs of proliferation. I can tell that for sure because we haven't really looked, but I really don't think there are any signs of apoptosis.
Great. Well, I think we reached the end. So well done, Kika. I mean, this was quite a valiant effort.
Thank you.
Your questions is quite a lot. So, I mean, I'd like to thank you for a really wonderful talk and wonderful answers to questions. So Kika's kindly agreed to have this recorded. So for anybody that didn't see it, can catch up, or if you want to sort of probe for the details, You can catch up on the YouTube channel. And yeah, I'd just like to thank you again, Kika. Really, really exceptional work. I really enjoyed it.
Thank you. Thank you, everybody who listened.
Thank you.
Okay.
And
All right.
see you all tomorrow again.
All right. Thank you. Bye.